Hello, everyone. My name is Tianyuan Liu, and currently I'm a PhD candidate from Long Track Doctor Network. And my supervisor is Dr. Anna Conesa, and I'm very excited to introduce our current work, Transcriptome Universal Single-isoform Control, which is a framework for evaluating transcriptome quality. So as you may know, long-read sequencing is a really amazing technology. It enables us to capture the full-length RNA molecules. So this enables us to do isoform level quantifications and also detect a lot of novel isoforms. However, the technology is not perfect. Compared with short reads, it has higher error rate and also lower throughput. And when you do multiple sample experiment, there are sample level variabilities, and also long-read sequencing have different particles like cDNA library particles and direct RNA particles. So it is very important to do this kind of benchmarking to user-defined transcriptome. And current benchmarking strategy may have some limitations. For example, the most frequently used one is RNA spec-ins. Using this synthetic RNA, user can basically understand the quality of their sequencing, but this synthetic control may not capture all the bias in long-read RNA-seq experiment. And the other way is to use simulation, but it's really hard to simulate all the bias and complexity in the RNA experiment. And the final way is to use TUSCO, which is a conserved single copy ortholog, but it's designed for benchmarking the completeness of a genome. So for transcriptome, maybe it will misinterpret alternative isoforms as gene duplicates. So therefore, we developed TUSCO, which is a selected list of genes with only a single isoform. And we use this list of genes to do the benchmarking. So we cross-referenced across several annotation databases. And after that, we check in short-read sequencing data to make sure those transcripts don't have potential novel splice junctions. And also, we check the transcription start site to make sure those genes don't have alternative transcription start site. And then we check across different gene expression databases to make sure those genes are universally expressed across several different tissues, so similar as housekeeping genes. And finally, we collaborate with gene code expert to do some manual filtering of this list of genes. So in the end, we have 54 TUSCO genes for human and 31 TUSCO genes for mouse. And in order to use this list of TUSCO genes, we integrated with the scanty software. So basically, we will compare the user-defined transcriptome against this list of TUSCO genes. And if the user-defined transcriptome can perfectly reconstruct the TUSCO selected gene, then we consider it as a true positive. If it partially reconstructs the TUSCO selected annotation, one consider it as a partial true positive. For example, here you can see there are some variations in the 5' and 3', and also maybe there is one missing exon in 5' or one missing exon in 3'. And if the user-defined transcriptome find a novel transcript with a novel splice site or novel splice junction, then we consider it as a false positive. And for the false negative is if the user-defined transcriptome don't have this TUSCO gene, then we consider it as a false negative. And using all those informations, we can create a radar plot to benchmarking user-defined transcriptome. And our first question is, we want to know if TUSCO can perform similar as RNA spec-ins like SERVs. So in order to validate our hypothesis, we used the Lau-Agas dataset. And the results shows that in human and mouse, basically TUSCO performs quite similar with SERVs, but here we can see TUSCO detect more partial true positive compared with RNA spec-in serves. And we think this is because TUSCO can capture more bias in RNA-seq experiment. And in order to validate our hypothesis, we use an RNA degraded dataset. And in this experiment, the researcher degraded the cell pellets under room temperature in different amount of time. And after that, extract RNA, add RNA spec-in sequence, and then do the sequencing. And we use TUSCO to do the evaluation. And here we can find that for the TUSCO benchmarking, we actually have a really high correlation with the RNA integrating number. But with the RNA spec-in sequence, we didn't find this correlation. So basically, this indicates that the TUSCO can capture these RNA degradation artifacts during the experiment, but RNA spec-ins cannot capture. And then they ask us a second question: could TUSCO also use to evaluate novel isoform discovery? And to validate and to do this, what we did is basically we delete the TUSCO selected annotations, and we added some simulated artificial splice site. And then we provide this simulated artificial annotation to the transcriptome reconstruction algorithm, and to see if those algorithms could reconstruct the original TUSCO selected annotation. And if the algorithm can reconstruct the original deleted TUSCO selected annotation, then means that it do a perfect job. And in this radar plot, it will have a bigger area coverage. And here we evaluate two algorithms. One is Bamboo and the other is FLIR, which is two most popular algorithms. And we can see that in Bamboo, when we provide the gene code reference annotation, Bamboo do a perfect job with a really with almost perfect coverage in this radar plot. But when we provide the TUSCO simulated annotation, you see it's the dashed line here. So basically, the performance reduced a lot. But if we look at the FLIR provide a more balanced result between the reference guided with true annotations and TUSCO simulated annotations. So this may tell us, so if we want to detect novel isoform, when there is already annotation in a specific gene loci, maybe we should use FLIR in this kind of use case. Okay, so I will make a conclusion. So basically here we provide a robust quality assignment framework called TUSCO, which use single isoform gene to do the transcriptome benchmarking. And this overcomes some limitation in synthetic controls. For example, synthetic control cannot capture the RNA derogation effect during the RNA extraction step. And also we integrated it with Scanty3, so you can easily use it to do your transcriptome benchmarking. Thank you for your attention.
Thank you.
A nice talk. So my question is that, so sometimes, you know, when you developing a method to identify in this different isoform, the real challenge is that, so there are different confounders. You know, some isoforms are quite really similar to each other. But in your study, if my understanding is correct, that is you are just have one isoform, right, for one gene, right? So that means you do not have that challenge for the different algorithm, right?
Yes. I mean, so basically, the basic idea we consider here is we select single isoform gene because they have very conserved splice site. And this is used as an internal control. And I mean, if one gene has several isoforms, then we don't know how many isoforms it actually has. So we cannot use it as a, you know, internal control to do this kind of benchmarking. So for example, if one gene have 5 isoforms, maybe they have another 5 isoforms that's not annotated.
Okay, thank you.
Over here, Tian. That was a really nice talk, and I really like the idea of having the internal control, based on the single isoform genes. You're starting actually with like the most complicated genomes, right? Human and mouse, and there are other genomes like the fly, maybe, that have a combination of not only just single isoform genes, but also genes with variable numbers of exons, like 1, 2, 3, 4 exons. And I wonder, when are you going to extend your method so that it would cover other species that people might be interested in?
Yeah, well, I mean, currently we only have human and mouse, and we can also cover other species, but the challenging thing is in other species, maybe we don't have very good short-read sequencing data. So we know that, those genes only have one isoform. So we are not sure about this in other species. So one way I'm thinking, maybe we can use some deep learning algorithm or to make this kind of, sequence to function things to make some prediction. But, it's hard when you don't have a lot of short-read sequencing data except human and mouse for other species.
Okay, let's thank our speaker. Thank you so much.
Thank you.